can happen without any mutations, but is upregulated in acidic environments, is upregulated in hypoxic environments, which oftentimes there is overlap, right? Especially in those hypoxic niches within a given cancer. On the flip side, within a cancer cell specifically, you can generate D2HG. So we're thinking chiral center, different enantiomers. We can generate D2HG if we have a mutant IDH, that's isocitrate dehydrogenase, another TCA cycle enzyme. If we have a mutant IDH, we can generate D2HG. Both of these substrates can inhibit alpha-ketoglutarate dependent enzymes. We can consider these TETs, Jumanjis, these are histone demethylases. So you're essentially generating molecules that look like alpha-ketoglutarate, and you are inhibiting alpha-ketoglutarate dependent enzymes, and you're learning about it specifically within the context of histone demethylases. So when we inhibit our histone demethylases, this keeps our DNA in a tightly bound form, specifically as it relates to this site, but this keeps our DNA in its tightly bound form. Right? When we do this, we prevent the transcription and translation of genes. This ultimately maintains a degree of stemness within cells. Right? This maintains a degree of stemness. What does stemness mean? It means undifferentiated, right? So preventing from further differentiation and sustained proliferation. And when we have that, it's very important in, for example, a stem cell if we don't want it to differentiate, or more importantly, a cancer cell which wants to prevent differentiation. Otherwise, it doesn't want to just become a tissue, it wants to keep on proliferating undisturbed. Right? So this maintenance of stemness is not just important in stem cells, but is also important in cancer cells which want to kind of go undifferentiated and continue to proliferate. So again, alpha-ketoglutarate can be reduced to make two different forms of 2HG, D2HG and L2HG. L2HG can happen canonically via LD, or can happen in LDHA or MDH1 and 2 without further mutations, and is upregulated by aciduria, or high acidic environments, or hypoxia. D2HG is generated via mutant IDH1 and 2, but both of these molecules ultimately inhibit alpha-KG dependent demethylases and prevent differentiation. D2HG is a much more potent inhibitor than L2HG. So D2HG is a much more potent inhibitor than L2HG. This is a diagram I stole from IPI, so I will give credit where credit is due. Yeah.
So do cancer cells want to stay methylated?
Yes, cancer cells want to maintain their stemness.
So they want to downregulate or upregulate the demethylation?
They want to downregulate demethylation. They want to keep the DNA untranscribed because it is implied that the genes that would be transcribed would be used in differentiation. So it's saying don't differentiate, let's keep those genes bound up. Yeah.
So do all cancer cells have that mutation?
No.
So
A certain class. This is like, so the thing with cancers is that like certain mutations, very much prevalent in certain kinds of cancers over others. L2HG and D2HG are very much implicated in gliomas, which are like brain cancers.
So then certain cancer cells would not want alpha-ketoglutarate because it would increase demethylation?
I would focus on like, just think about this within their respective like gliomas, right? Which is just consider this within the specific cancer pathway. As it is, again, still an area of research. So I don't want to overstep. Yeah. Any other questions about this? Okay, so before you guys go, let's do our barcode. Okay, and let's see responses. Okay, nice. On average, you guys are right. It's the first three, right, which are directly using. This is first. Okay, you guys are free to go. I will see you on Wednesday for the last class. Come prepared with questions. We'll do a topic by topic review.